Hi, I'm Mandy Colsey, and this is Case Connections for Unit 2. So first of all, I'm going to look at case number 1. So pyruvate kinase catalyzes the rate-limiting ATP-generating step of glycolysis, in which phosphoenolpyruvate is converted to pyruvate. Multiple isoenzymes of pyruvate kinase exist in mammals. Type L, which is found in the liver and kidneys. Type R, which is expressed in erythrocytes. Type N1, which is found in tissues such as muscle and brain. And type M2, which is present in self-renewing cells such as embryonic and adult stem cells. So the first question is, under normal conditions, describe all the ways in which pyruvate kinase is regulated in the cell. So first of all, pyruvate kinase is regulated by ATP. When a cell has a high ratio of ATP to ADP, the cell no longer needs to use glycolysis to produce more ATP. Glycolysis is halted altogether. Also, phosphorylation deactivates the enzyme, whereas low blood sugar activates the action of epinephrine and glucagon, which causes an increase in the level of cyclic AMP. The cyclic AMP activates the CAMP-dependent protein kinase, which phosphorylates protein kinase, deactivating it. Pyruvate kinase M2 type is also allosterically inhibited by alanine. Pyruvate kinase is activated by its upstream substrate, fructose 1,6-bisphosphate up here. And then type M2 is also allosterically activated by serine, as well as several other small molecules. So the next part of the question is, many types of cancer cells are predominantly glycolytic and overexpress the oncoprotein MYC. Interestingly, the classical oncoprotein MYC has been found to promote preferential expression of pyruvate kinase type M2 over pyruvate kinase type M1. The isoform M2 is characteristically found in a low activity state and is ineffective at promoting glycolysis when compared to pyruvate kinase type L or type M1. This logic seems counterintuitive for a glycolytic tissue. What is the rationale for this mechanism, and what other pathways may cancer cells be diverting substrate to? So the M2 form of pyruvate kinase is associated with proliferation in cells because it's more easily regulated than its isoenzymes. And although glycolysis is important to the cancer cells to produce ATP, cancer cells rely on their ability to be prolific. PKM2 can exist in a tetramer or a dimer form, and this dimer form is less active in the process of glycolysis. So it can be manipulated by cancer cells to control the action of the enzyme. Type M2 is able to be regulated to adjust to the needs of the cell. The inactivation of pyruvate kinase, which would occur right here in the process of glycolysis, is going to cause the accumulation of upstream substrates, and these can be used in other processes to allow cancer cells to rapidly reproduce. The upstream glycose-6-phosphate can be shunted to the pentose phosphate pathway over here. This will produce reducing equivalents in the form of NADPH, which is needed for biosynthesis pathways, as well as producing ribose 5-phosphates, which can be then used to synthesize more nucleic acids, which is very important in reproducing. So other intermediates from glycolysis can be used in lipid and amino acid synthesis as well. Those are going to help those cancer cells reproduce, in addition to helping to produce energy. So the next case is case number 4. In this question, a previously healthy 2-month-old female presents with jittery spells several hours after breastfeeding. Her mother reports she has not given the baby anything to eat other than breast milk since birth. The baby has only recently been sleeping for more than 2 hours between feedings, and that's when the mother reports these symptoms started. Laboratory results are consistent with hypoglycemia, which is low blood glucose, but are otherwise unremarkable. Epinephrine is administered, and there's no increase in blood glucose, but there is an increase in lactate. Physical exam reveals a liver edge 4 centimeters below the right costal margin, and percussion of the right chest and abdomen confirms hepatomegaly. Blood glucose levels increase after breastfeeding, but she cannot maintain blood glucose levels within a normal range during fasting. So based on this information, which of the following is the most likely diagnosis? We've got hereditary fructose intolerance, galactosemia, glycogen storage disease, and glucose 6-phosphate dehydrogenase deficiency. So looking at answer D, if the patient had a glucose 6-phosphate dehydrogenase deficiency, it would affect the pentose phosphate pathway. And this would lead to hemolytic anemia and jaundice because the red blood cells would have inadequate NADPH to reduce glutathione to eliminate reactive oxygen species. So choice D isn't quite what we're looking for. Those aren't the symptoms that she's having. Now in choice B here, if the patient had galactosemia, there would be a problem with the GALT enzyme, which means when she ingests galactose, which she does every time she breastfeeds, her body would accumulate galactose 1-phosphate, which is the phosphorylated galactose. And this would likely lead to hypoglycemia in the fed state because her body wouldn't be able to metabolize the galactose to glucose because of the block at that step catalyzed by the GALT. And you probably see some other symptoms with this as well, such as jaundice and vomiting, in addition to more consistent hypoglycemia. If we look at choice A, hereditary fructose intolerance, it can cause fasting hypoglycemia and hepatomegaly because the deficiency in allylase B keeps the fructose 1-phosphate from being cleaved into glyceraldehyde and DHAP. This causes an accumulation of fructose 1-phosphate and a decrease in the free phosphate that's needed in order for gluconeogenesis and glycogenolysis to occur in the fasting state. So this disorder seems less likely in this case because the baby is exclusively fed breast milk, which can contain fructose, but usually only a small amount. Hereditary fructose intolerance isn't usually seen in infants until formulas and foods that have higher levels of fructose are introduced. So this leaves us with answer C as the likely answer. So if we're looking at glycogenolysis and how it occurs here, with a glycogen storage disease such as von Gurk disease, an enzyme important to the body's usage of glycogen is deficient. So in the first few hours of fasting, glycogenolysis is the body's primary pathway to maintain blood glucose. As this infant is experiencing longer periods of fasting, she needs to be able to break down glycogen to provide a continuous supply of blood sugar, and something here is preventing that from happening. Because the glycogen is not being metabolized, it's accumulating in the liver, which is leading to the hepatomegaly. With the administration of epinephrine, glycogenolysis should have been stimulated, which should increase the level of glucose in the blood. This occurs because hypoglycemia stimulates the secretion of epinephrine, which acts on a G protein coupled receptor to generate cAMP, which stimulates the phosphorylation of glycogen phosphorylase to break down glycogen. The epinephrine administered is also going to increase the amount of pyruvate being synthesized, and it'll be in excess of what the citric acid cycle can utilize. So that's what's increasing the level of lactate in the blood. So especially if we look at the case of von Gurk disease, which is glycogen storage disease type 1, there is a deficiency of the enzyme glucose 6-phosphatase here, which means that glycogen can't be cleaved into glucose. And this will cause both hepatomegaly and fasting hypoglycemia, as well as lactic acidosis. So those are my case connections for this unit. Thank you.